Thank you very much, Jianping, for the kind introduction. Good afternoon, everyone. It's really my pleasure to be here. It's my first time here, and the weather is very nice. You get a really blue sky, just like California. So today, I want to talk to you about some of our work on stem cell engineering. The research in our lab is focused on cell and tissue engineering, and mainly in two aspects. On one hand, we are interested in how the biophysical factors, together with biochemical factors, to regulate stem cell function in vitro. And on the other hand, we're also interested in how stem cells participate in the regeneration and remodeling of tissue. In this case, we focus on vascular remodeling. So today I want to tell you two stories. One is the in vitro cell engineering, and the second will be the in vivo tissue regeneration and how stem cells participate in this process. We know biophysical factors exist everywhere in the body, from the system levels, organ, tissue, cell, into molecular level. In this example, you could see cardiomyocytes could generate contraction force, and this is through the Actin-Myosin contraction. So cells are the building blocks of our body. Now, they are not only generating forces, in this case, to fulfill the function of the heart. On the other hand, the mechanical forces or other biophysical factors could also affect the cell functions. So here are two examples in the literature. In the first example, this was the work done at UC San Diego. It shows some acute response of a cell to mechanical loading. If you apply a local force here, and here the signal is the SRC kinase activation, you can see the propagation of this signal in the cells within seconds to minutes. So this is an acute response. On the other hand, in this example, the work done at the UPenn, the interstitials group, shows that in the long-term effects of mechanical signal, in this case, the elastic modulus of the substrate could regulate stem cell differentiation into different lineages. For example, bone, muscle, and neuron tissues. So all of these are related to the stiffness of the substrate. For example, on a stiffer surface, the cells may differentiate to osteogenic cells. On a soft surface, it could be adipogenic and elastic cells, in the neuron cells. So there are many other biophysical factors. It's not just what I showed there, the local force loading or the elastic substrate. Here you can see if you have a cell in a tissue, there are many mechanical factors or biophysical factors. In the vascular system, we know the blood flow will generate shear stress on the surface of the cells. This can regulate many aspects of function in endothelial cells. In addition, there are soluble factors, of course, in the blood. In the substrate, we have dynamic mechanical loading due to the pulse of the pressure. In addition, there's some passive signal in the actual cellular matrix. For example, the stiffness of the substrate, as we have seen in the last example, and the micro and nano pattern of substrate. Because the substrate has a micro and nano fibrous structure because of the collagen elastin composition. The cell has to integrate all of these signals and then make decision either reorganize into certain structure or migrate, grow, or differentiate into different type of cells. So at a molecular level, in the past 2 decades, people have done a lot of work to show the mechanical signal or biophysical signals in general can regulate the mechanical transduction through the cell surface receptors, intracellular signaling cascades, and finally transmit it into nucleus to regulate gene expression in the cell functions. However, most of these processes are acute response. So you talk about the mechanical signal on the cell surface, and then within minutes to hours, you activate a certain signaling cascade, and finally regulate gene expression. And many of these are transient responses. Because when you remove the mechanical signals, this process will stop and go back to basal level. So when you talk about stem cell function, it usually involves the phenotype change and long-term change of the cell function, such as differentiation or transdifferentiation, which involves another important process, which is the epigenetic regulation. So epigenetic regulation happens in the nucleus. we know all of the genomic information is stored in the chromatin in nucleus. And the chromatin can be organized into a certain structure. The epigenetic regulation involves something physical, the physical organization, instead of the genome sequence. For example, in the neuron, the genes, the muscle genes may not be expressed because they are packed in different parts of the chromatin. In this case, will be the heterochromatin. In heterochromatin, the histone and DNA are highly compact. And this will not allow the access of transcriptional factors to regulate this gene expression. So these genes are turned off. In another structure in chromatin is the euchromatin. And this is loosely packed genes around histone. And these are subjected to transcriptional regulation. And the cells have the memory of the phenotype through the regulation of these two types of chromatin structure. And this regulation is through the modification of either DNA at methylation or histone modifications. This includes both methylation and acetylation. So how the biophysical factors or mechanical factors can regulate the epigenetic state of the cell is not known. So we are interested in seeing how mechanical factors can directly or indirectly regulate this structure, and then non-specifically or specifically regulate the cell phenotype change, such as stem cell differentiation or cell reprogramming. The epigenetic state actually has been described in this landscape, which is related to the engineering perspective. So here, each point on this landscape represents one epigenetic state. And this could be either stable or unstable. For example, if you have one cell, at this state it has high potential. For example, it could be pluripotent stem cell, embryonic stem cells. And these cells could differentiate into any of these lineages. You can see these are stable states in the valley, and this hill area is unstable. And when the cells differentiate, it could be a spontaneous process. It could be directed by specific factors or combination of both biochemical and mechanical factors. And it could be different cell types. And at the end of this valley, you reach a stable state. It could be mature cell types such as fibroblasts, could be cardiomyocytes, neurons, etc. So in the past, people think this process is a one-way street. The cells will be able to differentiate spontaneously from the pluripotent state into multipotent stem cells, and then unipotent and finally mature cell types. But in the last decade, people have shown that it is possible to move the cells around this landscape by doing cell reprogramming. So for example, you can use the Yamanaka factors, which 4 transcriptional factors, to turn a fibroblast into pluripotent stem cells. In the recent years, people also show it is possible to overcome this barrier to turn a fibroblast into cardiomyocytes, neurons, etc. So all of these were done with biochemical factors, or transcriptional factors or chemical compounds. So we don't know whether biophysical factors could also help this process to move the cells to change on this landscape or change the epigenetic state of cells so that the reprogramming process or the cell phenotype change could be easier. So we asked the question, in this case, how the biomaterials or mechanical factors could regulate this process. So in the past decade, we have tried to engineer different kind of scaffold materials with different micro and nanostructure. In this case, this example shows that we use electrospinning technique, which has been widely used in many labs, to make 2D or 3-dimensional structures, either with a certain micro pattern or with certain alignment of the fibers. And we have used this to engineer cells in tissue structure. For example, you could make the cells fuse into myotubes in a line in a certain direction, or enhance axon growth in a certain direction. So all of this could lead to some tissue engineering applications. And then we examine whether some of this structure could affect the cell reprogramming process. The hypothesis is that this micro nanostructure could regulate the cell morphology and then non-specifically changing the chromatin organization in the nucleus and finally enhance or suppress the reprogramming process. So we use this model of cell reprogramming developed by Yamanaka and many others. So in this case, we use fibroblasts either from the skin or ear. And then we add transcriptional factors in chemical compounds, same as what many other people have done. The difference is that instead of culturing the cells on a culture dish, we culture them on different biomaterials. For example, microgrooves or nanofibers with random or line structure, or the substrate with different stiffness, which has been done also in many labs here, including Jan Min's group. And we have shown actually all of these structures more or less have some effects on the reprogramming process. So here I just want to show you some examples with the microgrooves and nanofibers. So the protocol is like this. We first grow the cells and then infect them with the Yamanaka factors, which include 4 genes, OCT4, SOX2, KLF4, and CMYC. These 4 transcriptional factors. And then we see the cells on different kind of substrates and wait for 2 weeks. And during this period, the cells were cultured in a reprogramming medium. So what we observe first, which is not surprise, is that the cells will follow this micro pattern on the surface and change your cell morphology. And this has been observed for many cell types. You have cell alignment. At the same time, you see that nucleus shape also change. You have more elongated nucleus shape when the cells are cultured on these 10 micron wide microgrooves, which is just a little bit smaller than the cell size. And these are just some statistical analysis to show that not only the cell shape change, but also nucleus shape also more elongated. And then we examine the colony formation during this reprogramming, because when the cells, fibroblasts, are reprogrammed into pluripotent stem cells, they're going to form colonies. If you count the number of colonies for a certain area, per unit area, you can see actually there's a dependence on the microgroove surface. On the 10 micron microgrooves, it seems the reprogramming efficiency is much higher. This was a surprise, and we then did some follow-up study to see whether indeed these cells are reprogrammed. So we characterize this colony. So by staining, immunostaining, you can see indeed the cells express the pluripotency marker, for example, NANOC. And if you expand this colony, indeed they can grow on the regular culture dish. They're positive for the markers of pluripotent stem cells, such as OCT4, SOX2, NANOC, SSA1, etc. If you want to demonstrate they are indeed pluripotent, you could also transplant them into immunodeficient mice and show indeed they form teratoma. So in this case, we use 4 factors, OCT4, SOX2, KLF4, and CMYC. And we also try to reduce the number of transcriptional factors. Indeed, we found that if you use 3 factors, CMYC is an oncogene. In this case, we remove that. Just use OCT4, SOX2, and KLF4, we still have enhancement of reprogramming efficiency on the microgroove surface. So what happened exactly? How can the microgroove surface or this micropattern surface affect the cell reprogramming? And then we did some detailed study at the molecular level. It is known when you reprogram cells with transcriptional factors and chemical factors. For example, in this process, there are some epigenetic changes that can enhance reprogramming. People have done a lot of studies with different chemical inhibitors, etc., to show that if you have increased acetylation of histone 3, or you have enhanced histone 3 lysine 4 methylation, this will increase the reprogramming efficiency. So we examine whether the microgroove surface, in this case, can enhance these 2 signaling events and then regulate the epigenetic state. By using Western blotting analysis and immunostaining, indeed what we found is that here you can see the 10 micron grooves enhance the acetylation of histone and the dimethylation and trimethylation of histone 3 as well. So these effects are very similar to the chemical inhibitors. Here we have 2 chemical inhibitors. VPA is the histone deacetylase inhibitor that can enhance reprogramming, and TCP is the histone demethylase inhibitor that also enhances reprogramming. It was published a while ago. So when you culture cells on the flat surface and add the chemicals, the effects are similar to that on the 10 micron grooves. And with immunostaining, we also show that on 10 micron microgrooves, we have a change of nuclear shape, and also increase of acetylation and methylation of histones. So this suggests that these topographic cues could non-specifically change the global histone acetylation and methylation state, and that could regulate the epigenetic state of the cells. And then we compare whether the microgrooves could achieve the same effect as the chemical inhibitors we just showed here. So here you can see on the flat surface, the reprogramming efficiency is relatively low, and it is enhanced by microgrooves. And if you add chemical inhibitors, VPA and TCP, you enhance the reprogramming efficiency. And then when you culture the cells with both chemical inhibitors, you also reach similar reprogramming efficiency. So this experiment suggests that indeed the biomaterials in this case could have the similar effect as the chemical compounds and can replace them to enhance reprogramming of the fibroblasts into pluripotent stem cells. Another important finding in the literature is that if you don't use transcriptional factors, just use microRNA, you still can reprogram cells, but the efficiency is much lower. Of course, people have hard time to reproduce that. And here you can see the combination of two microRNA, 302 and 367. People have used that. That can reprogram cells in the presence of these chemical inhibitors. But if you remove these chemical inhibitors, basically you get no colonies. That means the cells are not fully reprogrammed. If you culture the cells on 10 micron grooves with these two microRNAs, we found that indeed you get some colonies positive for NANOC, which is the pluripotent stem cell marker. However, when we submit this paper for publication, review asks us to fully characterize these cells and make sure the cells are mature pluripotent stem cells. And we did the experiment. We found that, unfortunately, in this case, with microRNA and this biomaterial combination, we don't get mature pluripotent stem cells, and these cells are immature in the process of reaching the pluripotent state. So that suggests that we still need some transcriptional factors. And just the biomaterial alone, together with microRNA, is not sufficient to reprogram cells. Okay, so we have shown that indeed these topographic cues can change the epigenetic state globally. But the key is whether this happens at a promoter of these pluripotent genes that are important for the reprogramming. So to test that, we did chromatin immunoprecipitation with the antibody against the modified histone, either acetylation or methylation. And then we did PCR to test whether the promoter of OCT4, SOX2, and NANOC, these three major pluripotent genes, are regulated by these histone modifications. And here shows the PCR result. So you can see at OCT4 promoter, we only have modest enhancement of acetylation. So this could explain why the mechanical factors are not sufficient to reprogram cells, because these master pluripotent genes only is affected very minorly by the topographic cues. But for the other two genes, SOX2 and NANOC, you can see the acetylation is significantly enhanced at the promoter of SOX2, and also the trimethylation, but not dimethylation of the histone. And for NANOC, acetylation is also increased, and dimethylation is increased. So these biophysical factors could regulate the promoter modifications of some of these genes, but not all of them. So it's different from the biochemical factors. And then we did a lot of work. I'm not going to go through the detail. What we found is actually this modification of epigenetic state is regulated by HDAC and WDR5 upstream. And the question we ask is how these topographic cues could regulate these signaling pathways and then the epigenetic state. Of course, one obvious possibility is the topographic cues could regulate the cell morphology through cytoskeleton reorganization, in which could be transduced into nucleus to change the chromatin organization. So one possibility is, you can see here, the integrin links the extracellular matrix to cytoskeleton, and the cytoskeleton, actin cytoskeleton, for example, could be linked to the nuclear, lamin, nuclear matrix, and then linked to the chromatin structure. So if you disrupt the Actin-Myosin contraction with the chemical inhibitor blebstatin, you could see when you add a chemical inhibitor here, all of these change. For the histone modification, methylation, and acetylation, all are blocked. So this suggests that this mechanotransduction from the topographic cues to intracellular space is mediated by cytoskeleton. Without cytoskeleton, this doesn't happen. In addition, we also found something interesting. When the cells are cultured on these microgrooves, what we found actually there are some genes involved in the mesenchymal to epithelial transition, which is required for the cell reprogramming into pluripotent stem cells. This process is enhanced by the micropattern. So I'm not going to show all of this data, but I just want to mention that when the cells are cultured on nanofibers with either random or aligned structure, we observe similar responses. The cells cultured on the aligned fibers will have higher efficiency of reprogramming compared to the cells cultured on the culture dish or the random fibers. So these structures actually are quite different because microgrooves is kind of a regular spacing, regular depths. The depth is about 3 micron in this case. And here, with nanofibers, actually it's kind of a 3-dimensional structure. When the cells are cultured on these surfaces, the only thing common is the morphological change. So then we ask the question, is that possible if you don't have these topographic cues? Just grow the cells on the flat surface and give them morphological change. Will that induce the same kind of response as we observed? So we did this experiment. In this case, we used microstamping technique to pattern the islands of a matrix in either this perfect circular shape or elongated shape. In these two cases, they have the same culture area, but different cell shape index. Of course, this is a single cell culture. We cultured the cells and then did the immunostaining for the histone markers, in this case, the acetylation and methylation of histones. Indeed, we found that if you just have this cell morphological change, you're going to have nuclear shape change, and you have increase of acetylation and methylation of histone 3. So that suggests this mechanotransduction could be mediated by cell morphological change, cell nuclear shape change, which could be mediated by cytoskeleton. So here is our current working hypothesis. So we know the cells have cell-cell adhesion. Of course, they also have cell and matrix adhesion. And this cell adhesion is linked to cytoskeleton. And the cytoskeleton is connected to lamin, the nuclear matrix. And lamin matrix is connected to chromatin. It is interesting that people have found that the heterochromatin, which is a compact structure, is connected to lamin matrix around the nuclear membrane. And that's where the genes are turned off. And the euchromatin usually are in the middle of the nucleus. So one possibility is that when you change the connection between cytoskeleton and the nuclear matrix, this could non-specifically remodel the chromatin structure, either in the heterochromatin or euchromatin. And this could lead to some global change of histone modification or local change of histone modification, and finally change the epigenetic state to make the cell reprogramming easier or more difficult. So I also want to mention, besides our finding on the fibroblast reprogramming into pluripotent stem cells, we also found that if you have certain kind of substrate with different stiffness, you could regulate the fibroblast reprogramming into neurons. So this is a direct reprogramming process. That means the biophysical factors indeed could move the cells along this epigenetic landscape, change the epigenetic state, and make the cell reprogramming into pluripotent stem cells and other specific cell type easier. So that's what we found. I will not have time to show this part of the study here. So that's the first story on the in vitro cell reprogramming. The conclusion is that indeed biophysical factors could regulate the cell epigenetic state and then enhance the cell reprogramming. And now I want to switch the gear and show you some of the study on the in vivo tissue remodeling and regeneration. and how stem cells could participate in this process. I will use the example of blood vessel regeneration. So in the past decade, we have done a lot of study in vivo to test the role of stem cells in the tissue regeneration, and we have done studies in many types of tissues, for example, peripheral nerve, spinal cord, wound healing, blood vessel, heart, and even tissue regeneration. In the recent years, we have focused on blood vessel remodeling and repair. Of course, for tissue engineering, we know there are some paradigm. In general, a classical approach is to take some cell type and combine that with biomaterials and put in bioreactor to make functional blood vessel. So that has been done a decade ago. And then you have to go through some tissue preservation, and then you can do that clinically for transplantation. Of course, for some type of tissues, you can just use cell directly without scaffold. You can transplant the cells into the tissue, for example, for cardiac regeneration. You could also, for some case, just use a scaffold material without cells and repair certain cell type, tissue types. So here I want to show you for blood vessel regeneration, we started with an approach with the combination of cell and biomaterial, and finally we moved to a new approach with biomaterials alone. And this has some advantage to make the vascular graft available off the shelf, and also make the process much cheaper and faster. So blood vessel regeneration is very important. In the body, of course, we know almost everyone, when they're getting old, they're going to have some blood vessel diseases. For example, in a small vessel, this is coronary clogging. You also have, if you have carotid artery blood clogging, you can have stroke. In a peripheral vessel, you may have peripheral vessel disease, in some kinds tissue degeneration. And for dialysis, for some patient with kidney failure, they also use some shunt here between artery and vein, and this is artificial graft. So one problem for this is that when the diameter of the vascular graft is less than 6 millimeters, you have frequent clogging because of thrombus formation. So in the past few decades, people have developed different kinds of biomaterials. For example, EPTFE, Dacron, etc. These are still in the market, but these are all good for large diameter vascular graft, but not for small diameter vascular graft. So in the past 10 years, people have tried to use tissue engineering approach to enhance the biocompatibility of the biomaterials. In this case, to use some cell coating in a graft. You could use different types of cells. For example, at the beginning, people just use vascular cells, endothelial cells, and smooth muscle cells. And then in the recent years, people started to use stem cells because only these cells are expandable. And for the biomaterials, people have used synthetic polymers, and also try some native matrix as well to make vascular grafts. And here is just one example to show, indeed, you can use cell sheet to make artificial blood vessel with similar structure and property as a native tissue. So about 10 years ago, we started working on this project. For the scaffold, of course, you have many ways to make a tissue construct, and we selected this process, which is electrospinning, to make fibrous structures because this structure is similar to the native structure of blood vessel with the collagen elastin fibers in the vascular wall. This could also allow the drug delivery if you want to, because you have large surface area, and if you have cell infiltration, you will also enhance the remodeling of the tissue. We have tried many different kinds of polymers, polycaprolactone, PLLA, polylactic acid, etc., and they are copolymers. And we finally figured out a composition to make this kind of micro or nanofiber structure with the right mechanical property. In this case, you can see with the stress and strain curve, the graft mechanical property is very similar to the native red artery. So we could achieve that mechanically. And that's very important because when you implant vascular graft, you want to have a good match of a mechanical property so that the remodeling will happen in a favorite way. And at that time, what we're trying to do is to seed some stem cells into the graft to increase biocompatibility. So we choose to use mesenchymal stem cells isolated from bone marrow because these cells have some important properties. One is that it can be expanded, and you also have very low immune response because they are derived from bone marrow and compatible with the blood cells. Now, two properties related to the vascular cells. One is that these cells can differentiate into smooth muscle cells in the vascular wall. The other one is that it could resist platelet adhesion because it's compatible with platelet generated in the bone marrow. And we have shown this in a previous publication. So we made this graft, and it's very small. The inner diameter is only about 1 millimeter for the rat model study. And we seeded with mesenchymal stem cells and implanted into the carotid artery in the rat model. Here shows the two ends of the anastomosis sites, and this in the middle is the graft we made. And we did an extensive characterization to examine the patency, whether it's still open after a certain time period, the cell organization, and the matrix remodeling. Here I just want to show you one slide because it has been published a while ago. And interesting, what we found is that when you have mesenchymal stem cell seeding, here it shows the cross-section of the grafts. When you have mesenchymal stem cell seeding, the main effect is that you can prevent platelet activation in thrombus formation. You can prevent this neointima formation. Here it shows if you don't have cell seeding, endothelial cells, smooth muscle cells are staying all in a brown color. Here is the polymer surface. In this part, you can see this neointima formation and the narrowing of the lumen. And when you have mesenchymal stem cells, you have very nice endothelial cell layer. And underneath, you have a very nice smooth muscle cell layer. And you also have well-organized collagen and elastin synthesis. So in this case, what we found is mesenchymal stem cells could prevent platelet activation in thrombogenesis and make this graft more biocompatible. In addition, you could also enhance remodeling because it has some paracrine effect. They can release some growth factors in cytokines for the remodeling process. But one interesting thing we observe is that after 2-3 weeks, if you go back to examine what kind of cell types are in this graft, what we found is the transplanted human bone marrow cells are mostly gone. There are only few cells left in these grafts. And most of these cells are from the host, in this case from the rat. So you can use immunostainings to distinguish the cells from different species. So that suggests that indeed our body has regeneration potential. If you give them the right cues, you don't need to have all of the cells present in the graft. The key is to have the right chemistry to prevent the clotting at early phase. So then we switch to a different approach. We ask the question, is that possible just to use the biomaterial scaffold without cell seeding? In this case, we can make the graft available off the shelf, and it's much cheaper. And we try many different kinds of surface chemistry and see whether we can minimize the platelet activation and thrombus formation. For example, you can use mucin, a proteoglycan found in saliva, and also the joint fluid, which has you can see this kind of brush structure which can resist platelet adhesion. We also try something that can inhibit the activation of thrombogenesis cascade, for example, hirudin, a direct inhibitor of thrombin, or indirect inhibitor of thrombin, which is heparin. Here, I just want to show you one example. We use heparin to modify the surface, because heparin has some important properties. One, of course, is to prevent the thrombin activation and thrombus formation. And two, it has a negative charge. So it could allow the water retention and enhance the cell infiltration into the three-dimensional structure. In three, heparin also has can bind to a lot of growth factors. A lot of growth factors in cytokines has a heparin binding site, which allows us to immobilize these growth factors in a native structure to retain the activity. So our idea is this. Besides the inhibition of thrombus formation, what we hope is to accelerate this process of regeneration by recruiting some stem cells from the surrounding tissues. For example, if you can recruit smooth muscle progenitors from the surrounding tissues and recruit the endothelial progenitors from the circulating blood, it will help us to facilitate the regeneration of the blood vessels. So in this example, what we did was to use polyethylene glycol as a linker to conjugate heparin to the surface of microfiber or nanofibers. And then we immobilize the stromal-derived factor 1 onto heparin through the heparin binding sites. And this is just a small molecule with about 65 amino acids. And SDF1 is known as a factor that can recruit stem cells. In this case, we know that it can recruit both endothelial progenitors and smooth muscle progenitors. Indeed, if you stay in the surface of this scaffold, you can see this fibrous structure. All the surfaces are coated with SDF1. In the release profile, we can see if you don't have heparin, you're going to have a very quick washout by the flow. After 1 week, all the immobilized SDF1 is gone. But if you have heparin as a surface modifier, you can retain SDF1 for much longer period. Here shows in this red curve. So first we examine the patency. So indeed, if you have heparin, you could enhance the short-term patency. So compared to control, if you have 9 rats to start with, after 3 months, you only have half of the grafts patent. If you have heparin, you can improve the short-term patency. That means the vessel will remain open. But if you wait for 3 months, you can see a few more rats will have a clogged vessel. But if you have SDF1 immobilization, you have much better improvement in both short-term and long-term period. So the vessel, you can see here, you have some angiogenesis into the vascular wall, shows the integration of the graft. And if you look at the cross-section of those patent vessels, this is the implanted polymer scaffold. You have very clean lumen. So what happened? Does that really recruit the stem cells? So first we look at the lumen. We open up the lumen and then did a confocal microscopy. And what we show that, for example, at 2 weeks, you look at the graft at different locations, the proximal end, the middle part of the graft, and distal end. So what we found is no matter what you do to the surface, at the proximal end and distal ends, there's not much difference because there could be some cell migrations from the adjacent artery through the anastomotic sites. So what is different is in the middle of the grafts. So when you have SDF1, actually I didn't show the 1 week data here, you have more recruitment of endothelial progenitors. And these cells differentiate into endothelial cells because these cells was positive for CD34, and during differentiation at a certain time period they are positive for both markers as shown here. And for the case with control graft in heparin-coated grafts, you only have partial coverage of the surface on the luminal surface. So this suggests that could explain why the SDF1 immobilization could enhance the patency, because you have acceleration of endothelial cell coverage. So what about the vascular wall? If you look into the cross-section of this vascular graft, so this shows part of the vascular graft. What we found is if you have SDF1, you have more recruitment of a certain type of cells, and we characterize these cells and show they are kind of smooth muscle progenitors. And indeed, the recruited cells are not smooth muscle cells, they are not fully differentiated cells to start with, they are not fibroblasts. And these cells gradually express the smooth muscle markers you can see here with time increase. You have more of these cells. And if you isolate the cells at an early stage, for example, at 1 week, and for any of these grafts, of course, the number of recruited stem cells are different for this 3 situation. And the cells in culture, indeed, you can see they are positive for CXCR4 and CXCR7, which are the receptors for SDF1, suggesting indeed SDF1 will have some effects on these cells, possibly in vivo. They're also positive for another transcriptional factor, SOX10. We identify this as one of the adult stem cell markers. They're positive for some mesenchymal cell markers such as CD44 and CD29, but these are non-specific markers. And during when you have a condition to maintain the cells at undifferentiated state, and the cells are negative for the smooth muscle markers. They're negative for alpha-actin, calponin-1, and myosin heavy chain. But if you give them growth factors, for example, TGF-beta, or stimulate the cells with notch signaling pathway, you can see the cells can differentiate into smooth muscle markers, smooth muscle cells, positive for all of these markers. So indeed, we recruited some stem cells in vivo, and these cells may differentiate into smooth muscle cells during the remodeling process. So here shows the evolution of this concept. So the first generation of graft, which is still in the market, is for the large diameter graft. These are inert materials, in general hydrophobic, to minimize the interaction of the blood with the surface. However, with time, this is not non-fouling surface. You're going to have cell adhesion and finally thrombus formation. In the second generation, which is tissue engineering approach, with the cell-seeded graft, and this has a very good biocompatibility, but it will take weeks to make in vitro. And it's very expensive. It may take $20,000, $30,000 to make one graft, customized for patients. And this never reached the market, even though it has been done in different research labs and even in some preclinical trials. In the next generation, we think it will be the bioactive scaffolds. And these scaffolds will have the right structure in chemistry, not only prevent the thrombus formation, but also enhance the recruitment of stem cells, which can help the remodeling of the vascular graft. So here I want to make some more comments on the vascular stem cell we recruited from the surrounding tissue. So what we found is, not just for the large vessel, but also for the microvessel, these cells are recruited to help stabilize the microvessel formation during the tissue regeneration. So this is the hind limb ischemia model. So if you use a lineage tracing model, this is a transgenic mouse model, to label SOX10 positive stem cells, that's what we identify as the smooth muscle progenitors. In this case, all of the SOX10 positive stem cells, before and after differentiation, they'll be labeled in a red color, which is a red fluorescent protein. So here you can see in a ischemic muscle, if you have some vessel formation, these SOX10 positive cells could decorate around microvessels. Indeed, they are colocalized with endothelial cells. You can see endothelial cells are in the center and then surrounded by these red color RP positive stem cells. So in microvessel, these adult stem cells, smooth muscle progenitors, can also help the microvessel formation. And if you transplant these stem cells, we isolate the SOX10 positive cells from the GFP, the green fluorescent positive mouse, and we transplant into another wild type mouse. What we found is, indeed, these cells will form this structure around endothelial cells, which are positive for CD31. Now we are doing is trying to monitor the remodeling of microvessels in the animal model, in this case with the skin fold chamber. Here we show that with time, indeed you can see the recruitment of these stem cells. They can be activated and migrate towards the newly regenerated vessels. So the last thing we look into is the vascular disease. So one thing we found is that the vascular stem cells, or these smooth muscle progenitors, not only contribute to the regeneration and remodeling of the vascular graft, but they may also contribute to the disease development in the blood vessel. So in the past, here is the paradigm. So people have shown that if you have vascular disease, for example, atherosclerosis, neointimal formation, what we have is the deactivation, or the activation of smooth muscle cells and de-differentiation. In this case, what people have shown is that the mature smooth muscle cells could de-differentiate into immature smooth muscle cells and they become synthetic and proliferative, and finally contribute to this neointimal formation. However, one question remains to be answered is that whether the synthetic and proliferative smooth muscle cells could also be derived from stem cells. Because stem cells, when they differentiate, they could differentiate to myofibroblasts, which is exactly the same phenotype as the synthetic and proliferative smooth muscle cells. And these cells are positive for immature smooth muscle markers, such as alpha-actin, SM22, and caponin-1, but are negative for mature markers, for example, the smooth muscle myosin heavy chain. So we published a paper a couple of years ago to show a new model. In this case, we show that indeed in the blood vessel, there's a very small percentage of stem cells. These are quiescent stem cells in the normal vessel, but during the disease development or upon injury, the cells can be activated, and then the cells can differentiate into different cell types. In addition to myofibroblasts or synthetic smooth muscle cells, the stem cells can also differentiate into fat cells, cartilage cells, and bone cells, just like mesenchymal stem cells in other tissues in bone marrow. And these cells could contribute to the disease development in the vessel. That's why you could find some cartilage tissue in osteo tissues in the late stage of the disease. So the stem cells, these stem cells we found actually have the same kind of characteristics as we identify in the vessel regeneration. So one question is that during remodeling, what is the relative contribution of these stem cells and the smooth muscle cells? Because traditionally people think the smooth muscle cells are the major contributor of disease development. And that's the therapeutic target. However, there's no therapy developed in the past few decades to target these smooth muscle cells and cure this disease. So we did some study. In this case, we did a single cell analysis. So if you take the artery from mouse, rats, or even human tissues, and then you use enzyme digestion to isolate single cells, and you culture them on single island of extracellular matrix, and see how the cells expand. In this case, we can distinguish the effect of vascular stem cell in smooth muscle cells. So here shows the process. We collaborate with Ming Wu's group in electrical engineering at Berkeley to use these optoelectronic tweezers to take individual cells and put them into individual matrix islands. And with time, the cells will grow and expand. And what we found is interesting that the smooth muscle cells, which is a positive for myosin heavy chain, actually they grow much slower than the cells negative for myosin heavy chain. In this case, actually most of cells, if we do marker staining, we found stem cell or smooth muscle progenitors. So stem cells, even though they study with a very low percentage in the vascular wall, they could expand much faster than smooth muscle cells and become a major population in the disease vessel. That's what we found. Indeed, if you doing ex vivo study, if you take a piece of blood vessel and this is a cross section, you can monitor the vessel remodeling with time in culture, just tissue culture. And here you can see some green background of the matrix. And in this transgenic mouse model, SOX10 positive stem cells are labeled in the red color. And you can see within this 1 day, you can see some cells here, they could be expanded. And there are some activation of stem cells and migration of stem cells around in the vessel. So indeed, this small population of stem cells with injury, they can be activated and grow in the tissue. And then we did some in vivo study. In this case, these stem cells are labeled with SOX10 in LAC-Z with lineage tracing. So if you look into the native femoral artery, you have no color here. But if you have injury, here you use a white injury to denote the luminal endothelial cells. What we found is after 2-3 weeks, this part of vessel turns into blue color, positive LAC-Z, suggesting most of these cells actually in the near intima are derived from stem cells in this case. So in conclusion for this part of study in vivo, what we found is that vascular stem cells could participate in both regeneration and the disease development. And the regeneration process could be in the large vessel and the microvessels. And with that, I want to thank all the lab members, grad students and postdoc, and our collaborators. The names labeled in bold blue color, and these people are involved in the in vivo studies, and the people labeled in red color are involved in the in vitro studies I just presented. Thank you very much for your attention.
We're using sensors called inertial measurement units, and essentially it's a small box that inside of it has